And then we move on to our next talk, which is on a very related topic. And our next speaker will be Frederik Pahde, who is a PhD student at Fraunhofer Heinrich-Hertz-Institute in Germany. And he will speak about a paper that they published at the MICAI main conference this year, and it's called Reveal to Revise, an Explainable AI Life Cycle for Iterative Bias Correction of Deep Models.
Yeah, thank you. Let me share my screen.
Yes.
Okay, I think you should see my presentation now.
Yeah, perfect.
Perfect. Then, yeah, thank you for having me. So, as already introduced, I'm Frederik Pahde, and I'm a research scientist at the Fraunhofer Heinrich-Hertz-Institute in the Explainable AI group. And today I want to present our work that we published at MICAI with the title Reveal to Revise, an Explainable AI Life Cycle for Iterative Bias Correction of Deep Models. And this is a joint work with my co-workers, Maximilian Dreyer, Wojciech Zamek, and Sebastian Lapushkin. And our Reveal to Revise framework that we introduced has the task of, as the name suggests, revealing and revising model biases, so very related to the previous talk. And biases that we refer to here are data artifacts that are present in most of the training data sets that we use in machine learning. And one popular example is ISIC 2019, which is a data set for skin cancer detection. So it has images of moles of different classes. Some of them are benign, some of them are malicious. And examples are, for example, skin marker, band-aids, or rulers. And here on the slide, I show explanations for a VGG16 classifier that we trained. And those explanations are in form of heat maps, and they show relevance by input feature that is used by the model. So here we can see in the first example for skin markers that not only the mole itself is used, but definitely also a lot of the skin marker around the mole that is used by doctors to basically highlight suspicious moles. So this is an interesting feature for models to use. The next example is even more problematic. Here we have band-aids, colorful band-aids. So in the ISIC data set, there are a couple of hundreds of those images, but all of them are benign lesions. So this is one of those shortcut learning examples that was also mentioned before, that the model might use this as feature, that if there's a band-aid, then this is no cancer, it's benign. And this is, of course, extremely dangerous if we deploy those models in practice. And there are many more examples for different artifacts, including, for example, hair or skin color. So we introduced Reveal to Revise, which is a semi-automatic framework that detects those biases and corrects them. And on a high level, it consists of 4 steps that I will all address in a bit more detail in the coming slides. But those 4 steps are first the identification of model weaknesses, then the precise labeling and localization of artifacts, the model correction itself, and then the re-evaluation of the model after the correction step. So let's now look into all those steps in a bit more detail. First one, as mentioned before, is identification of model biases. So here, only a few methods exist in literature to automatically detect those biases. The first one that we are using is called SPRAY, which stands for Spectral Relevance Analysis, which was introduced a couple of years ago. And the idea is basically to have a pool of input samples. And for each sample, we make a prediction and compute those attribution heat maps. So we have a large number of local attributions, and then we can perform clustering on those attribution heat maps. And what this gives us is basically different clusters of explanations, which are clusters of prediction strategies employed by the model. And what we're looking for are outlier clusters, as here in this example. So we have one large blue cluster of normal explanations. And then we have one outlier cluster, which might indicate that the model is using a different strategy than usually here. And those could be related by data artifacts. Like here in this example, we can see that all of those samples in this outlier cluster contain those band-aids, and the explanations indeed focus on the band-aid instead of the mole itself. So that's the first method for revealing biases. The second that we used is called CRP, Concept Relevance Propagation, which is also a relatively recent method introduced in our group. which provides explanations that use human understandable concepts. And those concepts can be visualized by examples from the training set. And the idea is that each convolutional filter of one neural network represents one concept. And for each filter, we can retrieve the samples which mostly rely on this particular filter. And then going even further, we can also try to figure out which part of those input sample was used by the filter and mask out the remaining information from those input samples. And this gives us those really precise representations of concepts the model uses. So here we have two convolutional filters in our VGG network that clearly only focus on band-aids instead of other information. So this is also a way of figuring out that the model is using data artifacts instead of the model itself. So that's the first step. We revealed the model biases. Second step now is to generate rich labels. Because one problem with many model correction methods is that they require really dense labeling. It's really expensive to get those labels. Like, for example, precise localizations of those artifacts so that we can tell the model to ignore that information. So in our framework, we suggest a method to semi-automate this step by using so-called concept activation vectors, also called CAVs. And the idea of those concept activation vectors is to train a linear classifier somewhere in latent space where clean samples and artifact samples are linearly separable. So that's the assumption that somewhere in latent space that's possible. And if we train a classifier, then we can get the direction that points from clean samples to artifact samples. So we have one clear direction that represents the artifact in latent space. So we need the subset of labeled samples to train this CAV, but now we can apply it also on unseen samples to identify even more potentially artifactual samples. And bringing this even one step further, we can also use the CAV to compute explanations for the CAV predictor. So now we don't want to explain why a certain class is predicted, but we want to explain why the CAV thinks that the artifact so seed mesh artifact present. And this information can be used to extract the artifacts from the images and to have those really precise localizations or segmentation masks that are required for many correction methods. Which brings us to the next step, the model correction itself. Here we use three existing methods for model correction. Those are right for the right reason, Clark, and CDEP. By the way, I have all the references at the end of this talk. So if you're interested in more details for those methods, just check out the literature at the end. But the high level idea of right for the right reason and CDEP is to use the segmentation masks that we now semi-automatically extracted and fine-tune the model, telling the model that it is not supposed to use any information in those segmentation masks. So we penalize the model for using those input features. And this gives us a model that hopefully only relies on the model itself. Clark instead uses another approach. Here we only need the concept activation vector, the CAV, and then tell the model to ignore the direction in latent space that models the artifact for us. So it's a bit cheaper to apply Clark, but all of them need more information regarding artifact, whether the artifact is present or not. And this, after correction, will give us a heat map that only focuses on the mole and not on the band-aid, hopefully, if the method works correctly. And in our last step, we want to evaluate all of this. And here we suggest to use two different metrics. First one is the accuracy on clean and on poisoned data. To evaluate on poisoned data, we use the extracted artifacts to basically cut and paste them into samples from other classes. And if the model has learned shortcuts, for example, band-aid means it's benign, then the accuracy would drop on those poison data set if we wouldn't do any correction. So here we can perfectly compare clean and poisoned accuracy to see how much the model still reacts to those artifacts. The second metric that we use is the percentage of relevance on the artifact. Here we also use the segmentation masks to see where in the image the artifact is present. And then when we compute attribution heat maps, we can measure how much relevance is put onto the segmentation mask representing the artifact versus the rest of the image. And ideally, we want to bring this down to a really low number, like 0%. And putting this all together, this gives us our full framework, which we can now run also for multiple iterations. So having corrected one bias, we can look for other biases still left in the model and correct iteration by iteration. We also published code on GitHub if anybody is interested and wants to use this on their own data set or their own models. And looking at some results, we have successfully corrected the biases related to the 3 artifacts that I was showing in the beginning of the talk. On the first row, we have the vanilla model that was not corrected at all. And we can see that it still uses information regarding to all those artifacts. In the first iteration, we want to correct those skin markers. And if we look at those heat maps after the correction, we can see that the relevance on the skin marker was highly reduced. There's still some left, but it's reduced by quite a large margin. The other artifacts are still used because we haven't corrected for them yet. In the next iteration, we correct for the Band-Aid artifact, and we can see that the model now ignores the Band-Aid when making a prediction. And what is also important, it still remembered to ignore the skin markers. So this is also important in our iterative algorithm that it doesn't, so previously unlearned shortcuts are not learned again in the next iteration. And in the last step, we also unlearn the ruler. And if you look at the final model, we can see that it only uses the mole information for all those actually artifactual samples. So we successfully unlearned all those biases. If you're also interested in quantitative results, yeah, you can check out the paper. There we also have a bunch of numbers that I didn't want to show here. Yeah, which brings us to the end of the talk. So here we have all the references. First one is the paper that I just presented, but then also all the methods that we applied throughout our framework. So yeah, thank you very much for your attention. And if there are any questions, I'm happy to answer them.
Thank you so much, Frederik. So again, in the interest of time, we will have to keep the questions a bit short. There was one question in the Q&A that just popped in. Let me have a look. Okay, I'm sorry. This is it's too long for me to grasp so quickly. Maybe you can answer that one afterwards. But I have one question that I can quickly ask. Your methods seem to rely on the spurious features being linearly separable in the latent space, right? How critical does that assumption seem to be? Does that seem limiting in any way?
So there's a full line of research regarding concept activation vectors, which has found that if you choose the right layer, generally speaking, those concepts are linearly separable. So what we've usually done is that we use the last feature layer of the convolutional networks, where it's generally assumed that those concepts are really already separated, and we can linearly distinguish between artifact samples and non-artifact samples. If we have other artifacts like color or like simple shapes, it's probably better to also try other layers in the network, maybe earlier layers. But for the artifacts that we experimented with, we usually found that the last feature layer was a great choice and it worked better than for most of the other layers.
Mm-hmm. Interesting, thanks. Okay, so again, I'm sorry to cut this short, but maybe you, if you want, you can try to answer the other questions in the chat. And we will now have a super short break, and then we will be back in for 5 minutes. All right. Thank you again, Frederik.